We're good?
Yeah.
Okay, so today we're doing AP Lab number 7. This is genetics and information transfer. This in the past was called meiosis and mitosis. So how cells split into half to make normal other somatic cells, and how they split when trying to make gametes, which are eggs and sperm cells. Okay, so in part A, you are going to be playing with these little pop beads. You are going to be manipulating those little beads into different arrangements, showing your knowledge or trying to understand the concept of mitosis. Okay, it will tell you the steps and what to do with the beads. It has a centromere, it has some different colored beads, so you'll understand that. This was mom, this was dad, this is how they move around, etc. Part 1B, observe animal and plant cells undergoing mitosis, or in the stages of mitosis, using microscopes. There is a microscope at each station. At your station, you will see a fish, blastocele or blastoma slide. You will also see an animal, I think it's like an, I don't even know what it is. It's an onion or a, some type of plant cell slide. Both of those slides are already prepared for you. You're not staining, you're not creating the slide. You take the slide, you look at those cells under the microscope, and you're going to be jotting down some things. You'll actually be deciding what stages each of those cells are in, so you're going to be identifying those stages of mitosis. Okay, part 1C, garlic root tip stain and observation. I want to show you something cool. I didn't know if it worked, being my first time working through this, or doing this, setting this up. I took garlic, I put it into sand, I got the sand wet, I tried to make it as dark as possible, which means that these shoots coming up the top shouldn't have grown. However, it worked. Do you see these roots? Okay, these roots were not there a few days ago. Those roots were, they just had been created. So what do you think is happening in those roots? They're growing. They're undergoing mitosis. So these cells right here are going to be undergoing mitosis more than another cell that's not actively dividing and growing. So you're going to be working with these roots. We'll dust the sand off, and you're going to work with those roots. You're going to stain them and observe them. Part 2A, this kind of moves into meiosis. You're going to be playing with your pop beads again. You're going to be going through 2 divisions, meiosis 1 and meiosis 2. Hopefully, you will get the concept between how this meiosis stages, how the meiosis stages compare or contrast with these mitosis stages. Part 2B, Sordaria, this is crossing over. So with this step, I haven't done this too many times successfully, okay, but we actually have a plate, a petri dish that these are growing on. You will come with an inoculation loop. You will pick some of them up. You will kind of squash them and hopefully release these cells or this arrangement that will help us understand how much crossing over has occurred. If that does not work, I have little cards in the back, and I'll come around and explain how that works. But I have the cards made up for you. So if it doesn't work, we'll just use the index card, and it looks like what this should look like if all goes well. Does that make sense? Any questions on the steps here? Okay, for each one of these, you're going to be recording something, okay, in your lab packets that you're going to turn in. Okay, so we're done on the video.